Thank you again for the invitation. I'm really excited to be here and present some of our work. The last few years were really intriguing and exciting to me for a number of reasons. The pandemic triggered a number of new projects, and it allowed us as scientists actually to move a little bit outside of what you would normally do within your own sphere of knowledge and investigation. And this was not only a lot of fun and intellectually stimulating, but I think this is how we made actually a number of leaps and advances that I wouldn't have thought were possible before. So we are a stem cell lab. We work for over 2 decades actually now with what is called pluripotent stem cells. I will quickly introduce them to everyone. So pluripotent cells are very characteristic in 2 features. One of them is that they can, as long as they are stem cell, divide indefinitely in tissue culture. So technically, they are really immortal. The second key feature of pluripotent stem cells is actually that they can differentiate, like all other stem cells. But in this case, pluripotent stem cells, and this is why they are called pluripotent, can actually technically turn into any lineage that is known in the body. Right? All 3 germ layers, every single cell types. This makes it really interesting to study for a variety of reasons, not only because we have infinite supply of the stem cells and therefore can produce infinite amount of derived cells, but also the type of cells that we often don't really have access to. Another really important feature of pluripotent stem cells is that they are easily accessible to state-of-the-art molecular tools. Like, for example, CRISPR technology. Cell lines are easily genetically editable using CRISPR, and stable cell lines can be rapidly generated. Second, because we can make so many different cell types and so many of them, we can do a lot of molecular studies, including, for example, RNA-seq, single-cell RNA-seq with a lot of ease, then more sophisticated methods, like, for example, mapping the epigenome. I will show a bit of data on this using, for example, ChIP-seq or ATAC-seq. So this is our playground, so to say, scientific playground that we explored for quite some time, mostly interested in what factors drive really stem cells, the undifferentiated states, and differentiation. However, now this is almost 8 years ago. This is a good example of what happens in an anticipated way in the lab. My postdoc Sylvie, who studied at that time a new emerging field of pluripotency differentiation protocols, which were self-organizing entities. I will talk about this or explain this in greater detail in a second. And she connected with a researcher in the virology department at Mount Sinai, Michael Schotzertz, and at that time student Thomas Zwaka. And we started talking, and Michael told us about a new outbreak at that time of the Zika virus. And this was actually quite intriguing to us. And we felt, why not to combine both things, our ability to create really sophisticated, technically sophisticated systems in vitro that recapitulate key physiological aspects of a developing human tissue, in this case, the brain, and with Zika virus infection, and see what happens. And so this is what we did. This is the protocol that we developed. Others have developed at that time similar protocols. And it goes into the idea that what I just documented or showed to you, that pluripotent cells, provided the right signaling pathways, will differentiate into specific cell types. We don't really need detailed knowledge of a particular developmental milestones. In contrast, what we can also do is, just if we provide the right starting condition, allow the cells to differentiate spontaneously. We call this self-organization, and if you cluster the cells in the right way, in the right number of cells in the right tissue culture environment, like in this case, where we trigger differentiation into ectodermal lineages, we actually get structures that resemble a human brain. And this is quite a remarkable discovery of the last 10 years or so. That we don't really need all the developmental instructions that are incredibly complex. We just allow the system, once we reach this particular very specific point, we let the system go. And in this case, for example, it develops the human cortex with a relatively proper layering of the appropriate cell lineages, of the 3 lineages of the central nervous system, and it also includes actually stem cells. So we used the system, as I already explained. There were the mysteries of Zika virus infection at that time. It wasn't really clear why the infection would cause this terrible developmental defect, the microcephaly. And so we started developing this in vitro system where we can create relatively rapidly hundreds of these mini brains. By the way, they're actually pretty scary when you look at them. They really look like a little human brain in the bioreactor. And so at that time, there was the dominant idea that the infection somehow causes acute cell death, in particular of neurons. So there was some tropism towards neurons. But we were more interested because it is a developmental defect, also in the slightly longer consequences that lead to cell fate decision changes, and also to basically ask the question, what happens to cells that survive the infection? What are really the consequences that can explain in part the developmental anomaly, but also what are the consequences maybe even later in life? And so we took a system, these are cerebral organoids, brain organoids for short, that we differentiate. We established indeed that Zika virus readily infects certain types of cells within these brain organoids. All 3 lineages were infectable. All 3 neuronal lineages were infectable with Zika, but with different efficiencies. And the highest level of infection, actually, we saw with the neural stem cells, which makes sense because one prevailing idea was actually that the virus goes after the stem cells. They either stop differentiating, they're exiting the stem cell pool, and therefore there is not enough cellular material, simply put, to produce really the developing brain. And this is why we have the brain microcephaly. So while this was interesting, again, we were interested in longer-term consequences. So we profiled the epigenome of these infected human neurons in our brain organoids. And we found an astonishing number of changes in the methylation in particular. This is a global methylome analysis of a genome, in particular in key enhancer elements that were either hypomethylated or where we saw an increase in methylation, that all pertained to key developmental steps and also to genes that are associated with both neurological and later adult psychiatric disease. So this led us to postulate that perhaps one of the key factors that leads actually to the developmental defect after the infection was potentially a malnutrition, where not enough methyl donor diet was supplied. Remember, the microcephaly primarily occurred in areas in Brazil that were particularly poor or socio-economically deprived. And we think that perhaps a simple prophylaxis as folic acid may have helped to overcome some of the developmental defects that we saw. So this is something we proposed and would have loved to test further. Unfortunately, the virus disappeared entirely, or almost entirely. And so we were unable to continue the studies or the animal studies necessary to further test this hypothesis. However, then in 2020, the SARS-CoV-2 pandemic came about. We were immediately intrigued and interested in trying a similar approach as to the one I just explained to you, creating an organ-like structure that resembles key features of human physiology, and then looking specifically at infection with SARS-CoV-2. So for this, we again teamed up with essentially the same team, Adolfo Garcia Sastre, and some of the virologists in his units, in particular Chris White. And we developed a protocol to differentiate this time the human pluripotent cells into lung-like cells. In particular, what we focused on at that time, and it still remains true, were type 2 alveolar cells that seem to have the highest expression of ACE2 and are particularly sensitive to SARS-CoV-2 infection. So we optimized the protocol. In this case, we didn't really try to be as faithful as possible in recapitulating the natural environment and structure of the lung tissue, but primarily our optimization point was SARS-CoV-2 infectability. This led us to a system that, in contrast to the very faithful developmental recapitulation that takes several weeks to develop in vitro and is plagued with a lot of variability. After a few weeks, every of these structures looks slightly different. Our system, because it is so rapid, is actually highly reproducible and yields always the same amount of cells. The system itself is divided into two parts. I will mention them in just a second, but I want to highlight one particular aspect of the system that we think is important, and this is the rapid differentiation. We obtain the cells we are interested in roughly after a week in culture, in differentiation culture. So this was exciting to us, two researchers in our division, Marion Desjosé, and in Adolfo's division, Carles Martinez Romero, took on the project. We started optimizing, going back and forth, and came up with this protocol. And here I want to emphasize again why we do this seemingly complicated system where we first create primary like cells from an in vitro. And it has all to do with cellular tropism, in my opinion, because what is frequently overlooked when we just simply infect human cells, viruses to human cells, we typically take some kind of tumor cell or an immortalized cell line. But what is overlooked in this is actually that we have 3 levels of tropism that are highly relevant to the host response and the interaction between the virus and the host. First, there's the cellular tropism. Of course, as we all know, not all viruses infect off cells. In fact, there's highly specificity in terms of viral entry, but also all the necessary host factors. Second, the same applies to tissues. Not all tissues are the same, even having similar cell types to some degree. And there's enormous tissue tropism, again, that is recapitulated to some degree when we use these organ-like structures in vitro. And third, we have to also, and I will talk at the end about this, take into account that different hosts, like for example, human versus other species, may have a very different response to particular viruses, despite the same tissue being infected and the same cell types being infected. And here's some of our own data that documents the enormous differences in the different cell types, where immortalized cell lines, where we apply a particular drug to virus infection, don't really respond well. While our primary cells that were differentiated from pluripotent cells that are not immortalized will recapitulate the innate immunity in the most authentic way we can recapitulate in vitro, show a very significant dose response. So how did the protocol that we developed look like? Here we had to go back essentially to the developmental biology textbooks and remind ourselves that lung tissue actually is derived from the gut. It is a little structure, a budding structure from the pharynx, the trachea form, and then expands, starts budding, and creates all the sophisticated structure we associate with an adult lung organ. And so we had to recapitulate to some degree these processes. And just to keep it short, it essentially, the major milestones of this differentiation protocol are the development into the endodermal structure, which would be, in our case, an anterior pharynx-like endodermal structure, and then the induction of a differentiation process that leads to the formation of these lung-like structures that are budding. Once this is in place, the process of differentiation essentially takes place on its own and creates all the different cell types that we are interested in, including the highly ACE2 expressing alveolar cells. So this protocol was very efficient. We tested it early on, immediately, of course, with SARS-CoV-2, because this is what we're interested in. Here's the technical protocol for this. And what we found is this is single-cell RNA-seq analysis of the cells that were infected with SARS-CoV-2. And we find two things. First, high infectability. Essentially, 80–90% of the cells are infected. The cells not only infect, but produce high virus titer, similar or higher to virus cells, and they are produced by authentic human cells. So they likely recapitulate both entry, infection process, and exit of a virus in the most faithful way. We also find that there's a small subset, 10 or 20% of cells that are not infected typically, and they show a very significant innate immune response, while the innate immune response is suppressed in the infected cells. So this was very exciting. We started testing a number of drugs. This is how it looks in vitro and tissue culture. You see this cobblestone-like cells. This is the endoderm. And once we induce the differentiation of these lung-like structures, we see this process of clustering of these cells. This is the initiation point for the lung-like cells to differentiate. And then here on day 10, in this case, we see these large cells that have a lot of vesicles that are filled with surfactant. These are the type 2 alveolar cells. Again, we easily can infect the cells with SARS-CoV-2, and a variety of drugs that typically are known to affect infectability, viral processing, or exit of a virus, all in vitro in our system seem to recapitulate at appropriate concentrations the antiviral activity. The system was quite successful. We started collaborating with a number of labs. This is just a snapshot of the publications of the last 2 or 3 years where we used the system to confirm that whatever the researchers were asking, and I will show you 2 vignettes of the work in just a second, were successful. And highlight one collaboration here between a number of researchers across the globe, essentially, but in particular with Mount Sinai. This is with Chris White, a collaboration where we explored the efficiency of an inhibitor of an host factor. It's a protein translation inhibitor of a protein translation effector EF1 alpha, and drugs that specifically inhibit this translation aspect of translation. And again, it recapitulates, like in the standard cell line systems like Vero cells, 293 cells, it pretty much recapitulates all aspects, if not better than the other systems. But in this case, it's not an immortalized cancer cell line. It's a primary authentic human cell. Second example that I find quite interesting, this is a collaboration with Craig Weiland from Yale University, where they became interested actually in a unique aspect of virus infection, this regulatory, the gene regulation of the ACE2 receptor itself, and they find that a very specific chromatin remodeling complex, the SWI/SNF complex, actually is critical for expression of ACE2. And so by inhibiting this complex, they basically inhibit this process, and drugs that inhibit the SWI/SNF complex, typically associated, of course, are developed for cancer, actually quite effectively inhibit also viral entry and infection of SARS-CoV-2, here shown with our lung like cells derived from human pluripotent stem cells. Okay, and so because of this success and the significant interest in the research community for protocols like this, we teamed up here with the ASAP program, and the goal was to optimize further tissue culture system to make it more mid-level or high throughput level. And there were a number of considerations that we took into account, which were first standardization of the cell line. We homed in on a particular iPS cell lines that we developed in the lab that is very good for endodermal differentiation. And we showed that indeed we were able to reproduce all data that we use with the other pluripotent cells with this particular cell line. We also, in a more comprehensive way, did characterization studies that involved assays, in vitro assays for functionality of the cells, but even relatively mundane things like transcriptional profiling, where in the first major milestone, as I described to you, primitive endoderm, we see indeed differentiation into primitive endoderm and also the enrichment of classical lung type 2 cell markers that are associated exactly with a target cell type that we are interested in. So all of this was good. It didn't need much tweaking. What did need some tweaking was actually when we went from the higher content well format into a smaller content well format, is to optimize little things like the cell density itself and also the matrix that the cells adhere to, because it turns out that there are huge differences in how well the cells adhere. Remember, they have to be manipulated. In our case, actually, in many experiments, they have to be physically moved into a different facility for virus infection. And we're able to figure out optimal substrate and densities. And finally, what took us a lot of effort was actually also optimizing a freezing protocol with the idea that why would we have to go every time through the entire differentiation step, even though it takes only a week or 8 days? Why not to freeze the cells after these 8 days, QC them, and this way make them really immediately accessible to high throughput experiments if we batch freeze them. And this worked really well. We optimized a protocol using a controlled freeze down and special medium. And we are actively right now exploring how much this process accelerates really massive parallel infection of cells with SARS-CoV-2 and drug treatment. Furthermore, one goal, especially with ASAP, was to branch out not only into lung cells, but other pluripotent cells. And one target lineage was the blood lineage. We developed, again, a similar protocol as to the one I just described to you, but for hematopoietic cells. This is shown here. Again, it takes 2 steps, a mesoderm differentiation into bone marrow-like cells, and then or blood vessel-like cells, and then the final differentiation of hematopoietic cells. This worked. And so we have right now actually 3 protocols we actively are investigating for infectability, not only with SARS-CoV-2, but especially with other viruses of interest that we can claim are primary cells. And they include the endodermal derivative I didn't have time to talk about, which are hepatocytes, the blood lineages that we're able successfully to differentiate into macrophages, and then the last lineage that I showed in the beginning were neurons that we actively test for infectability with a variety of viruses. So what we want to do, as I said already, is expand the virus infection studies and also test the infectability with other cell types, expand more on the neurons, especially also what is of interest are cardiac myocytes, NK cells, and gut cells. And also find novel markers, of course, by when we differentiate them for optimizing infectability, and push further here the boundary we have into a 96 or 380 well format with all the modifications I just described to you before, where we found the optimal conditions that will allow us to do so. For the last 2 minutes, I just want to mention, because I was talking about host factors, and one important thing was actually the species. And we know already from the onset of the pandemic, it was thought to be a bat-borne coronavirus. And in fact, bats seem to be a particularly good host for a variety of different viruses. And we were very curious about this and thought, well, maybe we can develop a similar system as to what I just described to you, using human cells, differentiating them, for example, into lung cells. How cool would it be to do the same thing, but use, for example, in this case, horseshoe bats that are natural hosts for coronaviruses, differentiate them using a very similar protocol into lung-like cells infected with SARS-CoV-2 to see what are the major differences in these species in terms of response, because these bats are hosts without showing signs of a disease while we get sick. And so we teamed up with, and this goes back to what I said in the beginning, the fun in these international collaborations, especially in areas where you would normally not really branch out into. We actually connected with field researchers who study that particular bat species. This is how they used it with his postdoc. They caught us these horseshoe bats. And we received since also other bat species and developed a protocol to produce iPS cells. So we received them in the lab, applied tweaked reprogramming protocol to produce iPS cells, pluripotent cells from these adult bats. And this was successful. We developed protocols both from fetal tissue of bats that we obtained and also from adults through biopsies, wing punches, where we readily can produce iPS cells that show all pluripotency markers and most importantly can be differentiated into relevant lineages, including the lung cells that I showed you. And we are currently conducting SARS-CoV-2 infection studies, and they're actually quite remarkable in how different the cells deal with SARS-CoV-2 in response to the virus when we compare this to humans. And another lineage that we were successfully producing apart from lung cells was actually bat blood. And this also opens up an entire unknown box of how bats really deal with pathogens, in particular viruses, and how it compares side by side when we do the same experiment using human cells. What are really the key differences that may explain why bats are such a reservoir? And can we use this information actually to derive new ways, approach new drug targets for therapy in humans? This is just showing how different the bat iPS cells are in their transcriptional profile. It's quite remarkable. And so last slide, to summarize this, we want to use humans, but also other relevant species, make iPS cells, differentiate into relevant cell types, infectable, physiologically important cell types for the particular virus, phenotyping them like I described to you, infectability, transcriptional profiling, epigenetic profiling, that it will inform us then about new drug targets. And I see this as a bit of a loop, because drug targets develop in consortia, like for example ASAP, can be fed into the system and carefully compared, contrasted in terms of their efficacy, efficiency in vitro using these primary cells. With this, I want to thank in particular my lab. That was incredibly fun to work with, and with more difficult times during pandemic. But as you saw, we branched out in a number of collaborations, and I want to highlight especially the collaboration with Adolfo Garcia Sastre here at Mount Sinai and Chris White here at Mount Sinai. Thank you very much.